Hi, folks. How you doing today?
Good, how are you?
Good.
I'm okay. Thank you for asking. All right, welcome back. I hope you are well and that your families continue to be healthy. Um, I heard last week that one of my cousins had a bad case of COVID, and so sometimes these things sneak up on us. We kind of forget about them. So, all right. Well, welcome back, everyone. And let me congratulate you on getting past lecture exam 2. In general, the class average obviously for exam 2 was lower than exam 1. That's pretty normal for this course, where exam 1 gives you kind of a review of some basic ideas in biology, and then exam 2 really hits you with microbiology things. Okay, so if any of you want to walk through your exam together with me, I'd be happy to do that during office hours. Feel free to come talk to me about things that were on the exam that you struggled with. Okay, in general, the 2 questions where students most struggled was the gene expression question, where it gave you a snippet of DNA and you had to decode the RNA and then the amino acids of the polypeptide. That's one of those skills where if you make even one mistake, then you have errors in every subsequent step. And that's useful to think about in real cells, because if cells make a mistake in the DNA or RNA, you're going to get a mutated protein. And even a single change in a protein can be a problem. Look at sickle cell anemia. Sickle cell anemia is the result of a change in one single individual nucleotide of the gene for the globin protein, and that leads to a disease that we've all heard of. So that was a problem. And also the unit conversion work, as many of you know, was a problem as well. Please continue to practice those unit conversions. Please keep up with your skills. If you've learned them, don't let them go away. I will say this, that you will need your unit conversion skill for lecture exam 3. You will need it for lecture exam 3. Okay, so if you have questions about that, please seek out a tutor. Please get together with a classmate who seems to be doing okay in that topic. Please come visit with me during office hours. I'd be happy to help you. Okay, so let's get started here. We're going to continue our conversation in chapter 20. Chapter 20 is the antimicrobial drugs. Okay, and here we go. All right, gang. So let's talk. If you're following along in the textbook, the page number that I'm looking at in my physical book is 561. This is information that's around the foundation figure of 20.2. Foundation figure 20.2. There's a wall of text below that picture that says the action of antimicrobial drugs. Let's talk a little bit about that, okay? Mechanisms of actions for antimicrobial drugs. Here we're going to introduce you to maybe some new terminology, okay? When we talk about antimicrobial drugs, whether it be an antibacterial drug, an antibiotic, or an antiprotozoal drug, or an antifungal drug, or an antiviral drug, basically antimicrobial drugs will do one of 2 things. Okay, it is possible that a drug might kill the microorganism, right? You might kill the bacterial cell, you might kill the fungal cell, okay? If a drug kills the microbe, the drug can be described as a quote unquote cytal agent. Okay, a cytal agent. For example, okay, penicillin kills at least some species of bacteria by disrupting cell wall synthesis. So penicillin would be a bactericidal drug, okay? So we could have bactericidal drugs, we could have fungicidal drugs, we could have viralcidal drugs in the sense that, the molecule that we're calling a medication has the ability to completely disrupt the virus and knock out its reproduction. So it basically dies, even though, whether a virus is alive or dead is sort of a philosophical question without a point. So drugs can kill the microbe, and if they kill the microbe, they're referred to as a cytal with an ending CIDAL agent. Okay. Alternatively, you can have medications, you can have drugs that do not kill the microorganism, but instead sufficiently interfere with the growth and reproduction of the microorganism that effectively you are prevented from getting sick or ill from the microorganism. Okay. If a drug does not kill, but interferes, if it interferes with the growth and reproduction of a microbe, the drug can be described as a static agent. Static in the sense that the microorganism as a population of cells is not growing, okay, as a static agent. So we would change the terms above from bactericidal to bacteriostatic. Okay, we would turn fungicidal to fungistatic. And we would turn viralcidal to viralstatic. Okay, so if you're killing the microbe, you're a cytal drug. If you're suppressing the growth of the microbe without killing it, you're a static drug. Okay. All right. Let's talk about some strategies of various drugs. And here I'm going to give you specific lists of drugs. I'm going to give you specific names of drugs and give you some insight into how they work. Strategies used by antimicrobial drugs to kill or inhibit the growth of microbes. Okay. Well, one thing that can be done, and I'm following right along the majority of how your textbook is laid out. Again, the information is somewhere close to figure 20.2 if you're following along in your textbook. One thing that many categories of drugs will do is inhibit cell wall synthesis. Now, obviously, inhibiting cell wall synthesis doesn't make any sense when we're dealing with, cells that don't have cell walls. So for this strategy to work, we need to be dealing with a pathogen that has a cell wall. So most bacteria have cell walls, fungi have cell walls, that kind of thing. Okay. Inhibit cell wall synthesis. Okay. So this is a strategy that is carried out by several different drugs. Let me give you examples of drugs that work this way. Examples of drugs that use this strategy would include penicillin, and for that you see figure 20.3, figure 20.3 on page 562. Okay, so it tells you there that the inhibition of bacterial cell wall synthesis by penicillin is shown to you there. And you notice if you look at figure 20.3, it shows you there in the bottom right corner, it says the bacterial cell lysing, meaning rupturing open as the penicillin weakens the cell wall, right? That's basically how penicillin does its killing of bacteria. We have other antibiotics that also work this way. Vancomycin is one of them, and bacitracin. Okay? So all three drugs that I've just put here in your notes, all three of these drugs are antibacterial drugs. And again, from the common everyday usage of the term, they would be considered antibiotics. Okay, all right. So let me ask you in the chat window, folks. Here we go. Do you have a tube of bacitracin in your medicine cabinet at home? Okay, because it's a very common antibiotic that you can get in over-the-counter creams and things like that. If you have a child that falls outside and scrapes his or her knee and you want to clean it up nice and good and prevent any possibility of infection, put a little bacitracin cream on there, okay? So just pointing out that that's something that many folks do have in their medicine cabinets at the house, okay? All right. So let's see here. Next category, letter B. So one strategy used to kill microbes is to inhibit cell wall synthesis. I'm specifically giving you examples here of antibacterial drugs. Okay, another category for killing microbes, and we're going to stay with bacteria a little bit here, is to inhibit protein synthesis. Okay, that is on my book, it's page 561, where you see a figure heading, no, excuse me, where you find a heading called inhibiting protein synthesis. Okay. You recall that bacterial ribosomes are described as 70 S particles, right? Eukaryotic ribosomes are 80 S particles. So that means there's a structural difference in the composition of a bacterial ribosome compared to a eukaryotic cell ribosome. And that difference is enough to exploit a strategy of successfully killing bacteria with leaving, generally speaking, human cells alone, okay? So remember that bacterial ribosomes are 70 S particles, and eukaryotic cell ribosomes are 80 S particles. Okay, you think about it, when we have any kind of pathogen around that would make us sick, well, theoretically, I could kill any pathogen, right? If I was able to put anything toxic into the body that would kill the pathogen. But there's a good chance that that toxic substance would actually kill the person at the same time, right? So, I could cure any case. I could cure any case of flesh-eating bacterial infection if I was allowed to put hydrochloric acid on the surface of the skin. But obviously, you as the patient would really not appreciate that, would you? So the point here that I'm trying to make with these ridiculous ideas is that when we're talking about drugs, we have to have a drug that does two things. It has to kill the pathogen and it has to leave our own cells alone, right? We don't want toxicity. A lot of times we do have toxicity, and those are side effects of medications that, we probably have all had side effects of various medications throughout our lives. So let me give you examples of drugs that fit into this category. Okay, I'm just going to copy this text here, put it down here. Okay, examples of drugs that use this strategy. would include chloramphenicol, streptomycin, and tetracycline. Okay. I'd be willing to bet that most of you have heard at least one or two of these drugs, right? You probably have heard of tetracycline, chloramphenicol, streptomycin. I bet you've heard of many of these things. And so what I want to do here is go into a little bit more detail about how these medications work. We're going to share with you some medications that there's something meaningful we can say about it. Okay, in other words, we have enough scientific understanding of how the drug works that we can tell you something about it. Okay, so let me ask a question in the chat window. Put a drug on the market to treat people, is it a requirement that we, meaning scientists, understand how the drug works? Is that a requirement? Okay. All right. All right, gang. So thanks for participating. I deeply appreciate it. I really do. And let me say something that might be shocking to you. Of all the things that are required to put a drug on the market, being able to explain how the drug does what it does is not a requirement. It is not a requirement. Okay. Okay. To get a drug on the market, let me tell you what is required. And I'll just put this in the chat window since I started here. Place a drug on the market, you must provide two pieces of information. Okay, number one, you have to provide safety information to show that the drug does not have horrendous side effects in the human body. And two, you have to show that the drug has some kind of positive therapeutic value, that it will do what you want it to do, which is kill the bacteria, kill the fungus, kill the virus, kill the protozoa, whatever it is that's making you sick. Okay, so once again, in the chat window, to place a drug on the market, you must provide two pieces of information. One, safety information that there's no horrendous side effects, and two, show effectiveness of the drug. So if you look at that, okay, part of that explanation does not include a deep understanding of how the drug does what it does. And that is my point. There are many, many drugs being sold out there on the marketplace today for which we know very little about the actual mechanism of how the drug does what it does. I mean, scientists might be able to give you a long-winded guess, but there's a lot of things we can't really tell you. All right. So to get a drug on the market, you need to show safety information and effectiveness, not an understanding of how the drug does what it does. If we needed to understand how a drug does exactly what it does, which I know at first glance seems like it'd be useful information, there'd be far fewer drugs on the market that could be sold to people. Okay, so let's get back here. Chloramphenicol. And what I want to do is just give you a statement or two about how these drugs do work in terms of what we scientifically understand about what they do. Chloramphenicol, which you see in figure 20.4, and I'll reference this in your notes here. Chloramphenicol binds to the 50S subunit of the bacterial ribosome, right? So that would be the large subunit of the bacterial ribosome, and it interferes with peptide bond formation. right? So if we take away the ability of a bacterial cell to manufacture protein, then it cannot manufacture enzymes to sustain all the necessary chemistry that the cell needs to live, and the cell will therefore die. And the fact that chloramphenicol selectively binds to the 50F subunit of the bacterial ribosome, while largely not binding to the equivalent large subunit, the 60S subunit in a human cell ribosome, means that generally speaking, we have very few to no side effects for taking this medication. All right, the next drug is tetracycline. I'm going to put this one next in my list. Okay. Tetracycline has the ability to interfere with transfer RNA binding to the ribosome. So if the transfer RNA whose job it is to bring amino acids to the ribosome cannot complete that task, once again, you interfere with protein synthesis because the ribosome cannot grab onto the amino acids it needs to assemble the growing protein. Interferes with the tRNA binding, to the ribosome. Okay, this prevents the ribosome from making protein. Okay. Next, we have streptomycin. Streptomycin is a pretty neat example here. There's two things I want to say about this. Streptomycin, keep wanting to put an E on there, but there is no E on streptomycin. Streptomycin alters the shape, it alters the conformation, it alters the three-dimensional geometry of the small subunit, the 30S subunit of the ribosome, so that the codons of information in the messenger RNA are actually misinterpreted, and that leads the bacterial cell to manufacture junk protein. Okay. Streptomycin alters the shape, technical word here is conformation, of the 30S subunit of the bacterial ribosome, causing the ribosome to misread or misinterpret, if you prefer, the codons of the messenger RNA. So the bacterial cell makes junk protein. What's interesting about streptomycin is two things. First off, the bacterial cell is going to make a protein which does absolutely nothing useful for the cell. Okay, it's kind of like going to the store and buying something you didn't want or didn't need. What was the point? Okay, but in addition to that, not only are you making junk protein, but think about it. You have to actually waste energy to make junk protein. So the cell continues to waste its precious energy reserves, its ATP stock, to make something that's completely useless to the cell. Okay. Bacterial cells make junk protein and waste precious ATP fuel doing it. Okay. All right. So again, getting back to this idea that, when we're trying to treat a pathogen inside the body, we would prefer a drug that is very selective in killing the pathogen and doesn't really cause harm to non-pathogenic cells, meaning our own cells of the human body. Okay, that's a useful feature of drugs, right? Okay, so let me state that here in your notes. is important for drugs / medications to kill pathogens inside the body without influencing the metabolism of human cells that make up the body. Okay. Whenever you have a drug that does have a side effect, whatever that side effect might be, maybe maybe the drug gives you headaches or whatever it might be. Whenever you have a drug that gives you some kind of undesirable side effect, it is almost always because the drug is not completely inert to the human cell. I'll say it this way in your notes. Whenever side effects from drugs do exist, it is usually because the drug is changing the physiology of the human cell in some subtle way. Okay. And so let me ask this question, and I'll put this question in your notes because it's worth considering during your study time. Okay. Why should we, meaning students of science, why should we be suspicious that some antibiotics to kill bacteria might be capable of poisoning our own human cells? Why should we think that antibacterial drugs might be a problem for human cells? What do you think, folks? Would you have any thoughts on that? Would you like to take a guess at it? Certainly. Okay. Any takers out there? Anyone want to be brave and venture a guess forward? Okay, Chelsea. Chelsea, you make a very interesting statement there. Take a look, folks, at Chelsea's comment in the chat window. She says, because we, meaning humans, require good bacterial cells to function. That's a good point, right? So Chelsea, if you've ever been on a treatment for anything with an antibiotic and you've had some diarrhea or anything like that as a consequence, we know that a lot of times we get diarrhea because the antibiotic is not only killing the bad bacteria, but it's killing off the good bacteria that live inside us, and that produces, some of these nasty side effects. So Elena, you talk about the chances of affecting our normal cells and destruction of them. Okay, I want to stay with that. I'm going to come back to that in just a moment. Fatimata, you say they kill the good and bacteria, and we need the good bacteria to survive. Also, a very reasonable and good statement, Fatimata. So you're correct, ladies, that antibiotics will kill the good bacteria off at the same time as killing the bad bacteria off, and we might have some side effects because of that, like diarrhea, constipation, things like this. That's all true. Those are good points you brought up. But take a look at my question. Why should we expect that some antibiotic might be capable of poisoning our own human cells? Not to draw this out too long, but here's what I'm suggesting. I'm going to give you a hint in the chat window to see if you can come up with this on your own. What organelle inside your cells is related to bacteria? Come on, I know you know it. Okay, Aubriana is the first to answer if someone will run with it. Thanks, Aubriana. You're absolutely correct. Remember the endosymbiotic theory that we discussed for lecture exam 1? The mitochondria in human cells and in plant cells, chloroplasts, are of an evolutionary relationship with bacteria. Let's put it plainly. We think that at least some of our cells, organelles, are actually primitive bacteria that live inside our cytoplasm. And so what I'm saying is that if you have a drug like an antibiotic that can kill bacteria, if that drug is capable of entering your human cell, and why shouldn't it be? Because after all, our cells don't have a cell wall to block it. All we have is a plasma membrane. So in some cases, small quantities of antibiotic will actually penetrate our plasma membranes, enter our cytoplasm, get into our cells. And if the mitochondria are really bacteria, and that drug works by killing bacteria in any number of various different ways, doesn't it seem reasonable that that antibiotic might disrupt the proper, normal, healthy functioning of mitochondria? In fact, that has been observed throughout scientific research. We've observed that happens, okay? So how shall I say this in your notes? Remember that certain organelles like mitochondria are really bacteria. Therefore, if an antibiotic works by killing bacteria, if the antibiotic enters a human cell, there is a decent chance it may disrupt the physiology of your mitochondria. And that can have toxic consequences. Many diseases that we are aware of seem to have some kind of connection to abnormal mitochondrial functioning. Okay. So if you ever have a side effect with the drug and say, I just feel a little bit off, I don't know, because it's going to depend on a million different variables, but it's not unreasonable to suggest that maybe something that's happening is your mitochondria are being disrupted by that medication. Okay. So Elena, yes, mitochondria are the powerhouse of the cell, right? Absolutely. So maybe while you're on the medication, you just have no energy, right? You feel exhausted. You figure it's because I'm fighting the infection. That's probably true, but it could also be because you're poisoning your mitochondria in your cells while you're fighting the infection. Okay. All right. Thoughts, questions, gang? All right, let's continue here. All right, so our second strategy for killing or interfering with microbes is inhibiting protein synthesis. Let's see, the cell wall thing was figure 20.3. We'll get that into your notes. All right, next bullet point here is the letter C. For letter C, I'm going to say inhibit nucleic acid synthesis. All right, question in the chat window. Here it comes. Is it easier to heal someone or hurt someone? I mean, we all know we've been alive long enough to appreciate that life is very fragile at times. It is all too easy to hurt someone in some consequential way, which they never recover from, both physically and emotionally, right? Okay. So the point here is that there's a whole variety of different ways of killing microbes. And that's kind of what this lecture is about, right? We want drugs that selectively kill microbes that enter our bodies and leave our cells alone for the most part. Well, in the category of inhibiting nucleic acid synthesis, we do not have a lot of medications that are very good in this category. And the reason for that is because the process of manufacturing new DNA during the process of DNA replication, which you have familiarity with now from chapter 8, or the process of making RNA during the transcription process, which is also chapter 8, it turns out that the mechanisms of making DNA and RNA are so important that Mother Nature has really not changed them a whole lot between a human cell and a bacterial cell. What I'm saying is there are significant similarities between how a bacterial cell makes DNA and RNA and how a human cell makes DNA and RNA. So we do not have a lot of drugs that are very good at selectively killing, let's say, bacteria by inhibiting their nucleic acid synthesis, because more often than not, that same drug will screw up our own cell's ability to make new DNA and RNA. Okay? So while interfering with the ability of a cell to make DNA and RNA is an excellent strategy to kill the cell, unfortunately, bacterial cells and human cells share such a common evolutionary relationship for this important process that we have been very hamstrung as a scientific community to develop medications that can do this in a safe way in the body. There are not many useful drugs that can safely disrupt the synthesis of DNA and or RNA of the pathogen in your body without also making you sick. Okay. All right. So there are a couple here that I'm going to mention as drugs that are not fantastic drugs to have. I mean, if you're a patient and you're on these drugs, you're not going to be a terribly happy camper because there are some side effects. But there are some medications that can be somewhat tolerated by the human body and have been used historically in medicine to treat certain diseases. So examples of drugs in this category. Examples of drugs that use this strategy. Let me give it to you this way. First, we have rifampin. Rifampin. Okay. Your current book does not have any pictures of rifampin or anything like that. Rifampin is mentioned in table 20.3 when you look at the nucleic acid synthesis inhibitors of that table. Okay, so rifampin is an example here. Rifampin is also known as rifampicin. Okay, this medication interferes with the ability of a cell to manufacture messenger RNA. Okay, and so students of history might appreciate that rifampin has been and even today is used commonly, particularly in underdeveloped nations, to combat outbreaks of tuberculosis. Okay, rifampin is commonly used to combat tuberculosis, which you know is caused by the bacterium, Mycobacterium tuberculosis. The reason that rifampin is used to treat tuberculosis is not because it's a tremendously nice drug to put into the body. I mean, if you're on rifampin, you're not going to feel well, okay, because it does have poisonous side effects. But the reason rifampin is still used today is because as a drug, rifampin has very good ability to penetrate deeply into tissue. It has good tissue penetrating ability. Okay, and so you know, when a physician prescribes a medication, an antibiotic, let's say, to treat an infection, the doctor has to take into account where the infection is in the body, right? If the infection is in the body, in the bloodstream, then you would give that medication at a certain concentration level. If that infection happens to be in the inner ear, which is a lot harder to treat because the medications don't get into the inner ear as effectively as they get into the raw bloodstream, then you need to typically crank up the amount of drug you give to the patient and or increase the amount of time the patient is on the medication, because you need time for that drug to get into those far-reaching, difficult to penetrate tissues. So rifampin is used primarily because of its good tissue penetrating abilities. Okay, rifampin, of all the medications we might have to fight against Mycobacterium tuberculosis, rifampin is pretty good at getting into your lung tissue because that's where the microorganism, that's where the bacterium is hiding when you have tuberculosis. So rifampin is good at penetrating lung tissue. Okay, so, you know, even saying that, you know, if you're a tuberculosis patient, you could easily be on antibiotics for 6 months or longer, depending on what you're dealing with, okay? All right, so rifampin is one category of drug that fits into the strategy of inhibiting nucleic acid synthesis. It does kill cells in general by interfering with messenger RNA production. There is another category of drug, another type of drug that fits into this category, and it is known as quinolone. Quinolone has the ability to inhibit DNA replication. Okay, and what I'll say here is just as a way of, you know, kind of some interesting trivia. There's a drug on the market that is a close chemical cousin to quinolone, and I think you might be more familiar with it. This chemical cousin of quinolone is quinine. Quinine, and as you know, or as you may know, both quinolone and quinine are used to treat malaria. Used to treat malaria patients. Malaria, as you probably also know, is commonly transmitted through the vector of the mosquito, but the mosquito is not the actual pathogen. The pathogen is a protozoan that infects the body of the mosquito. Okay. The etiologic agent, which means the agent that causes the disease, is a protozoan by the name of Plasmodium falciparum, which is commonly transmitted by mosquito. Okay, so what I'll say here, folks, just as a way of re-summarizing our notes, okay? We're talking about strategies used to kill microbes. So far, everything I've talked about has been antibacterial, right? So up here for inhibiting cell wall synthesis, penicillin, vancomycin, and bacitracin are all antibacterial drugs. Inhibiting protein synthesis, chloramphenicol, streptomycin, tetracycline, these are all antibacterial drugs. When we get down here, letter C, rifampin is an antibacterial drug. Quinolone is an anti-protozoal drug, okay? An anti-protozoal drug. Let me highlight that so you don't miss it during your private study time. Okay, thoughts or questions right now? Now, folks, as you read through chapter 20, obviously you see, if you have your book open, you see, table 20.3, antibacterial drugs, table 24.4, differential grouping of cephalosporins, table 20.5, antifungal drugs, antiviral drugs, antiprotozoal drugs, anti-helminthic, anti-worm drugs, okay? Folks, we don't have time to go through all of that, and certainly I don't want you to memorize that. That's not a useful time of your energy. I would like you to read through the tables. You're probably going to see many things you've heard of before, but don't try to memorize it. In terms of testing purposes in some future exam, the things I'm going to hold you responsible for are largely the drugs that I'm currently putting in your notes, okay? So if the drug is in your notes, it is testable. If the drug's not in your notes, then I really don't expect you to go out of your way to memorize information about it. All right. Well, that's the third strategy, inhibit cell wall synthesis. Let's go to the third strategy, was inhibit nucleic acid synthesis. Excuse my French there. Fourth strategy of interfering or killing microbes is to inhibit the synthesis of an essential metabolite. Okay, so this sort of begs a question here, and I'll put this into your notes, but we should also answer it. So I'll put it in the chat window. What is a metabolite? What is a metabolite, folks? What do you think? Okay, thanks, Elena. An enzyme actually could be a metabolite. If we take a broad definition of metabolite, it could be an enzyme, okay? Okay, all right, thanks Chelsea and Ireland for contributing. Ireland, I like a lot what you said there. Okay, you said an end product of metabolism, okay? Thanks, Yasmin, very good. Metabolite sounds like it should be a fancy word, right? I mean, it sounds fancy, but it's really not that sophisticated of an idea. Folks, a metabolite is simply any molecule that is manufactured by the machinery of a cell. That's all it is. A metabolite is any molecule made by the machinery of the cell. Okay. So if you think about, Elena, you said enzyme. An enzyme is a protein. Well, is a protein made by a cell? Of course it is. It's made by a machine called a ribosome. So an enzyme fits the definition of a metabolite. Okay. So a metabolite is any molecule made by the machinery of the cell. In other words, any molecule that's manufactured by the chemistry of the cell that supports the cell's metabolism. Okay. I guess we should probably, I'm going to put another question in here. What is metabolism? What is metabolism, right? We're going to be heading towards chapter 5 next in our course sequence. And chapter 5 is microbial metabolism. So I'm giving you a little of a foreshadowing here. Well, what is metabolism? Okay, thanks for those that are trying out. Okay, so Elena, breakdown of a substance. Taylor, breakdown. Okay, many times students do emphasize the breakdown capacity of metabolism, but understand that metabolism includes chemical reactions that make things. They don't just always break things down, right? So the point I'm trying to make here is metabolism refers to all the chemistry inside a cell. All the chemistry inside a cell is metabolism. Whether that chemistry leads to the breakdown of something, whether it leads to the synthesis of something, whether the thing that I'm making is energy, it's all metabolism, folks. Say metabolism is a generic word that says grab all the chemistry that's happening inside the cell and understand it. That's metabolism, okay? Metabolism refers to the sum total of all chemical reactions inside a cell at any moment of time. The sum total of all chemical reactions inside a cell at any moment of time. So again, that's chemical reactions that break things down, and it's also chemical reactions that build things, okay? So, all right, we're talking about inhibiting the synthesis of an essential metabolite here. And what I will use is, hold on a sec while I find the figure here, is figure 2013. Figure 2013, let's put this in your notes. Okay, so figure 2013 might look a little scary because, you know, there's actual chemical structures there, right? Don't worry about that. I'm not going to beat you up with that on an exam, but I want you to understand the basic idea here that's taking place. Okay, so a little bit of background so that you can understand this, okay? All right, okay, so in bacteria, when bacteria need to manufacture DNA and RNA, or when they need to manufacture protein, they need a special substance, and that substance is called folic acid, okay? So here's the basic idea. Bacteria manufacture folic acid. Bacteria manufacture folic acid, and here's the basic idea in a flow chart. So bacteria are going to take in some food source. I'll do it this way. Bacteria are going to take in some food. As they nibble on the food, as they break it down, as all the chemistry of the metabolism takes place, the bacteria will use the building blocks of the food to manufacture something called PABA. Okay, so PABA stands for para-aminobenzoic acid. I don't care if you know that. I'm just telling you that's what it is. PABA stands for para-aminobenzoic acid, and PABA is a metabolite, meaning that bacteria manufacture it. Bacteria do not absorb it from the food, they manufacture it from the food, okay? So up here where I say bacteria manufacture folic acid, it's important that you appreciate that bacteria do not absorb folic acid from food, Okay, bacteria don't do that. They make it, they don't absorb it. So bacteria take the food, they manufacture this metabolite, which we're calling PABA. They then use the PABA to manufacture folic acid, which is another metabolite. Okay, the folic acid is then used to manufacture, and if you're looking at figure 2013, you can kind of guess at it yourself. They use the folic acid to manufacture nucleotides and amino acids. Okay, they use folic acid to manufacture nucleotides and amino acids. Okay, and so folic acid and any precursor that's needed to make folic acid, such as PABA, are absolutely important to allow the ultimate synthesis of nucleotides and amino acids, right? So if we don't have nucleotides and amino acids, well, then the cell can't make DNA, it can't make RNA, also can't make protein. That sounds like a disaster for a bacterial cell, and it would kill a bacterial cell if they cannot make the folic acid. Okay, so nucleotides and amino acids are essential to manufacturing DNA and RNA and protein. If you can't make DNA, if you can't make RNA, if you can't make protein, the cell is dead. That's it. Game over. You're not gonna, the cell will not survive. Okay. So just to give you a little bit of background here to wrap this kind of story up, human cells, okay, as an example of a eukaryotic cell, human cells do not have the enzymes capable of manufacturing PABA and folic acid. Our cells can't make it. So that means our cells absorb it, okay? Human cells do not manufacture folic acid, but do absorb it from our diet. Okay, so you can see this critical difference here. Bacteria make folic acid. They do not absorb it from their food. Human cells do not make folic acid. Our cells absorb it from our diet, whatever we're eating. Okay, and so let me ask this in the chat window. And this might mean a little bit more to the ladies in the room rather than the guys, and that's okay. But guys, you might be married, or you might be married someday to a gal. So let me ask this. Here it comes. Why do prenatal vitamins contain large quantities of folic acid? Why do prenatal vitamins contain large quantities of folic acid? Think about the conversation we've laid out over the past 3 minutes. Okay, right? The idea here is that if you're going to start a family, right, and the mom is pregnant with an unborn child, that unborn child's body is developing. That unborn child's body is going through many, many rounds of mitosis and copying the DNA many times simply to build the overall anatomy of that unborn child. And so to do that, that unborn child needs ridiculously large quantities of nucleotides and amino acids. Well, to do that, to make the nucleotides and amino acids, the cell needs folic acid. Our bodies, our own cells can't make it, so we need to absorb it in our diet, which is why it's important for a woman carrying a child to have plenty of folic acid in his or her diet. Many times they don't, which is why pediatricians, family doctors will put a woman on prenatal vitamins to support the growth and development of the child. And as you say, Rachel, in the chat window, it's extremely important for brain and neuronal cell development to have these building blocks. Okay. All right. So absolutely critical that, that's an important thing. If ladies, if you ever are thinking to have a child, go get some prenatal vitamins for your baby. Okay. So we're back here talking about inhibiting the synthesis of an essential metabolite. Here's the background I've just outlined for you. So what medical science has discovered is two different medications that can screw up the production of the nucleotides and amino acids in the bacteria by blocking production of either PABA, by production the conversion of PABA to folic acid. Okay. So I'll put this note in here. Okay. Now let's see. Maybe I'll put it down below here. Okay. Number 3, drugs that use this strategy would be the drugs in your book, sulfamethoxazole, that's a mouthful, and trimethoprim. Okay, trimethoprim. Both of these drugs in bacteria are capable of blocking the chemical pathways that lead to the conversion of PABA into folic acid. These drugs work at slightly different steps in the conversion process, as you can suggest from looking at figure 2013. But the upshot of this is that both drugs interfere with the conversion of PABA to folic acid. And so here we do have an example of drugs that are pretty selective for killing bacteria in the body and work pretty well with that subset of bacteria that are sensitive to these medications. Okay. There's not too much toxicity from these drugs in the actual human body because, again, our own cells are not concerned with manufacturing PABA or folic acid. We simply absorb it and go along our merry ways. Okay. So these drugs are both antibacterial drugs. Okay. So if we look at our laundry list of drugs that we've listed so far in our conversation, everything's been antibacterial except for quinolone, which is antiprotozoal. Okay. All right, hang in there, gang. We're doing okay. Let's keep going here. So strategy D was inhibit the synthesis of an essential metabolite. Strategy E is next. Strategy E is damage the plasma membrane. Okay. Damage the plasma membrane. And I'll put this down on a fresh page here. Okay. We do have some drugs that fit into this category or strategy, if you will. It generally work by changing the permeability of the plasma membrane to various substances, which then might promote death of the cells through osmotic principles. Okay. So let me give you some examples here. Drugs in this category often work by changing the permeability of the plasma membrane and altering the flow of water into or out of the cell. Okay, so okay, thanks for, I didn't see your comment there, Rachel. I think you're talking about the prenatal vitamins. So yeah, okay. All right. I honestly say I've never tasted one, so I appreciate hearing that. Okay. So let me give a silly idea in the chat window. Have you ever heard of death by chocolate for people that are crazy about chocolate? I mean, I don't know about you, but my wife is a chocoholic. I mean, she has to have chocolate every day. It's crazy. Okay. Okay, Chelsea. Yeah, stay away from that chocolate. So what I'm getting at here is when you damage the plasma membrane and you alter the flow of water into or out of the cell, I'm basically talking about death by osmosis. Okay. Not death by chocolate, but death by osmosis. And so we have examples of drugs that fit into this category. I want to tell you about two of them. One is polymyxin B. Polymyxin B. And polymyxin B is a drug that targets bacterial plasma membranes. Okay. I won't say any more about that. It's just another antibacterial drug, but you might have, you know, depending on your medicine cabinet, you can get polymyxin B and over-the-counter medications as well, over-the-counter creams. So you might have, you know, an antibacterial cream in your medicine cabinet that has polymyxin B. The one that I want to focus a little bit more on is amphotericin B. Okay, so we have polymyxin B and amphotericin B. Please don't confuse those, right? Polymyxin B targets bacterial plasma membranes, and amphotericin B, I'm putting into your notes now as a singular example of an antifungal medication. This targets fungal cell plasma membranes. Okay, so this is an antifungal cell. This is an antifungal drug. Okay. All right. And it's a little bit curious. I can tell you a little bit about how amphotericin B works. Amphotericin B has the ability to bind to a special substance that you find abundantly in fungal cell plasma membranes called ergosterol. Okay. So let me ask you another question in the chat window. Okay, if you happen to know it, maybe you do, maybe you don't. Okay, so amphotericin B binds to ergosterol molecules that are abundant in the plasma membrane of fungal cells. Okay, I'll put here in parentheses that human cells do have ergosterol, but in a much lower amount. Okay. So I'm highlighting the fact that fungal cell plasma membranes have a large quantity of this thing called ergosterol. Human cell plasma membranes, not so much, which is why amphotericin B is fairly selective for fungal infections and can be used fairly effectively and safely in the human body. The answer to my question in the chat window is as follows coming through. Another name for ergosterol is vitamin D2. Okay, vitamin D2 is ergosterol. Okay, so you might be familiar with vitamin D3, but in this case, we have vitamin D2. Okay, so polymyxin B is an antibacterial, right? It is an antibacterial drug. Okay. All right, so amphotericin B as a medication binds to ergosterol molecules in the plasma membrane that changes the permeability of the cell to the flow of water. And then depending on the surrounding environment, you can either have too much water enter the cell, overinflate the fungal cell and basically burst it, or you could potentially have water leave the cell, deflate the cell, and damage the plasma membrane of the cell that way. Either way, you have too much water entering or too much water leaving, you kill the fungal cell. Okay. All right. Okay, gang. Wow. Okay, any questions right now? Okay. All right, so next topic. So we've talked about these various strategies. The majority of drugs I've given you in your notes are antibacterial. I gave you one antiprotozoal drug, which is quinolone, and I gave you one antifungal drug, which is amphotericin B. Now I need to talk to you about some antiviral drugs. And, we could give you another long list of drugs that exist, but that wouldn't do much in the way of helping you understand how an antiviral drug works. So I'm not going to get it finished today, but I am going to start the conversation here in just a couple minutes to tell you about an example of an antiviral drug that's used to treat primarily herpes virus infection. Okay. So let's see here. So I'm going to be talking to you. I'm going to change the topic here now, specifically to antiviral drugs. I want to give you a case study in acyclovir. A case study in acyclovir. Okay. All right. So I need to give you a little background information or maybe remind you of some things that are very important for you to understand so that you can appreciate the acyclovir story that I want to tell you. Okay, acyclovir as a medication is used to treat herpes simplex viruses, which typically give human beings genital herpes and cold sores. And this drug is also used to treat herpes zoster virus. Okay, so we have herpes simplex virus, we have herpes zoster virus. Herpes zoster virus, as you know, causes chicken pox and unfortunately later in life shingles. So if you know anybody that has ever had shingles, you know that that's a pretty unpleasant thing. It's very painful to have shingles. Okay. All right, folks. By my clock, I have 1:45, so I really can't tell you this story in like 60 seconds. So I'm going to stop here. Okay. Do you have any questions right now? Okay. Folks, thanks for your time and energy today. We'll be back on this chapter next time. Yes.
I have a concern, but it's not about the topic we just treated.
Sure, Falashati. Do you want to talk to me after class?
No, I'll prefer to say it right now.
Okay.
It's about the exam that we did, the second exam.
Yes.
What I notice is, as for me, what I notice is that maybe majority know how to type very well and faster. But as for me, I'm not that fast in typing. And I think we have much of the essay and there was no much time for us. And so I would suggest maybe if we'll be having much of essay in the exam, then we should have much time to do that. And also I would like to ask if there is any kind of extra credit for the course.
Okay. Just for the benefit of everyone, there is not extra credit. I do not give extra credit. Afalashadi, we should talk a little bit after today's class. Can you hang on for like 3 minutes after class?
All right.
Okay. All right. Are there any other questions about content that we described today from chapter 20? Okay, folks, have a good day. We'll see you on Thursday. We'll finish chapter 20. We have lab on Thursday. So I'll see you then, okay? Take care.
Thanks, Dr. Allen. Take care.
Dr. Allen, I have a quick question. This is Elena.
Yes, Elena.
Hey, so I submitted my lab assignment early this morning. And I'm not quite sure, I've been having problems with this whole submission because it's in a PDF form, but my computer went out, so I was using somebody else's computer. I want to confirm that you received it so that I have enough time to look into that.
Yes, I have it, Elena. Thank you for sending it.
Okay, and it's visible and everything, right?
Yep, I'm looking at it right now. It looks good.
All right, great. I'd like to possibly chat with you about the unit conversion, the two questions you had talked about in the beginning of lecture. I really can't do it right now, but I'll make some time and come in for office hours sometime this week to discuss that. Because I know Falashadi just said with the whole essay and everything, I feel the same, that possibly it's a lot of information to have to kind of absorb and get my thoughts straight and everything. And I think I had a good idea of it, but just the timing kind of just threw me off, with the whole conversions and everything and trying to situate everything. When I looked at it, I'm like, but I knew that, like I knew it, so just we'll touch base at that.
Okay, yeah, come see me, okay?
Okay, sure will. Thank you.
All right, sure.
Bye-bye.
Bye-bye. Okay, hi, Falashadi.
Hi.
I definitely hear what you're telling me about the typing issue. Would this be something where you would feel comfortable getting a learning accommodation for it?
I don't understand your question. Like someone to teach me how to
Um,
type?
Well, no. What I'm saying is you're stating that you don't have enough time to type information into the computer. Is that what you're telling me?
Yeah, I'm not that fast in typing because I'm an international student.
Okay.
And where I'm coming from, we don't make use of the computer that much. We make use of the notes and our pen. We write. I'm fast in writing, but in typing, I'm very slow. I'm just getting used to it
Okay.
when I got here.
Okay. Absolutely. I understand what you're telling me. And so what I'm suggesting is if you can make a case with our student access office that you need extra time for exams because of your typing skills and the fact that you don't feel confident with typing, if you can make a case with them, then they'll get in contact with me and then I can legally give you more time on an exam.
You said student access?
Yes, hold on a sec. Let me bring up your syllabus in the course, okay? Because this information's in your syllabus.
Yeah, I think I saw it had a down part for accommodation, but I don't know. This can be part of what you're talking about. I don't know.
Right. And honestly, I don't know either, but it's something that it's the only way that I could give you more time on an exam. So hold on a sec. All right. Let me share my screen with you. Okay. So yes, at the end of your syllabus, it talks about students in need of accommodation, okay?
Yeah.
So what I'm suggesting to you is not that you are disabled,
Yeah.
but I'm suggesting to you your lack of typing skills puts you at a disadvantage. Is that fair to say?
Maybe, but I don't really know. So that's why I'm saying I don't know if it's part of what you're talking about for disability.
Right. So, just to let you know that I had students complete this exam in 58 minutes.
Yeah,
Okay,
I could see the way you type. That's why I have to raise the concern. Like
right,
you type faster, but I can't do that.
right. And so what I'm saying is that to give you more time, I need some kind of legal justification for doing that. And you just telling me that I can't type fast is not justification. This is something that has to go through the Student Access Services Department. So if you are willing to do so, then you could reach out to Vicki Van Heitz and tell her that you're struggling in microbiology because you don't type fast enough to get the information into the computer, and see what she says about that. She might say, okay, we can deal with that and we can get you more time on the exam. Or she might say, that's not something we can deal with and we can't get you more time on the exam.
Okay, in case she says that's not what they can look into, because what I'm actually talking about, that's why I had to raise the concern in class. Like maybe I will have some people like me that have the same issue, but I'm so surprised. Maybe I'm the only one. So what I'm just trying to maybe like plead or suggest is maybe you can just give us more time, or maybe the essay will be a kind of reduced or be limited because of the time. I don't know. I don't know what can be done from your own side.
Right. Well, I need to think about it,
Because
Shadi.
before I could really question, think of the answer I'm putting down,
Mhm.
time has gone. Then I want to type it again. Just like the unit conversion, I have to solve, I have to do this. Okay. And that one was even fair enough. Like we don't have to put everything on the system. We have to write it down and post you the pictures. So that was fair enough for my side. But the other ones,
Okay.
like I have to type them, they were quite much for me for me to go back.
Let me ask you this. Would it be easier for you for essays like you just had on the exam? Would it be easier for you to simply write your answers out on paper and send me a picture of it rather than typing it into the computer?
Yes, it can
Okay.
be easier.
Do you think that you would have been able to complete the exam in 1 hour and 25 minutes if you would have been able to just write out your answers and send them that way?
I think maybe probably it should.
Okay.
And I think maybe if it was reading, maybe I would have done better than what I'm having.
I understand what you're telling me, but obviously you're taking an online course right now, right? I mean,
Yeah.
this is all through the computer, and that's the deal. However, I would be willing to consider a future exams. I'm not going to give you more time, not unless you get documentation from student services. But I might be willing to allow you to write your answers on paper and send me the picture rather than typing it into the computer. If you think that works better for you, then we could make some accommodation to do that.
All right.
Okay.
Yeah, thank you so
Um,
much.
So, if you would like to do that for next exam, please just send me an email to remind me of this conversation, because there's some
Definitely.
technical issues, like if you send me your answers through email, let's say, and you don't actually type your answer in the class marker, then the grade on class marker is never going to accurately reflect what your true grade is. But of
Yeah.
course, it will reflect, I will have your correct grade in my grade book, but you won't be able to see it in class marker because you're not putting information into class marker.
Oh,
You see the difference?
yeah, I got
So
you.
in other words, I would have to grade what you send me through email and send you your grade back through email for your exam grade, which is doable. I can do that.
Okay,
Just so you know that if you went on the class marker, what class marker told you would not be accurate.
okay,
Okay.
okay, that'll be fine.
Okay.
Thank you so much.
All right. So, I know this is a concern and I don't want you to damage your grade because of a typing issue. So just send me an email before next exam, just to remind me about this, and we'll work that out. Okay.
All right. Thank you so much.
Okay. Have a nice day.
And you too. Bye.
Bye-bye.